All right, sorry about that. So this is our first video on cell communication, and it's going to be kind of a mini unit. It's only going to last like 5 or so days after the cells unit, and it has to do with, what do you think? Cell communication or cell signaling in the cell cycle. So we're going to talk about how cells can communicate with one another, how they can respond to stimuli in their environment and how they can reproduce to make new cells. So the very beginning here, we're going to talk about how cells communicate, either with one another or with themselves, because believe it or not, a cell communicating with itself is actually really important. And so this picture at the beginning really sums up what I wanted to explain about cell communication. So firstly, um, think about it as making a vending machine transaction. You are going to communicate with that vending machine about what you want by selecting something specific on the touchpad and by inserting money. All right? So you need to communicate with it in a way that it understands you. You can't, for example, use this. And this is an old gold certificate. It's the largest one ever made for $100,000. Even though this would be, by today's value, be worth way more than $100,000, is meaningless to this vending machine. You can't even buy a bag of chips with it. So the communication is more about the receiver of the communication than the sender of the communication. And that's one thing we're going to see repeated a lot when we talk about cell communication. So there is kind of like a cellular internet. So within multicellular organisms like ourselves, cells have to communicate with one another to make sure that they're doing the right things at the right time. To do this, they utilize what's called a signal transduction pathway. This is like a series of steps by which one cell, a signal on a cell's surface is converted into a specific cellular response. Right? And signal transduction pathways are very similar in all organisms, even similar between us and like yeast cells. So there's a big utility in learning about them because they're applicable for almost all of life. There's kind of three different broad types of communication methods. There's cells that are in direct contact, so touching each other, communication between those. There's communication or signaling between cells that are close by, which we call locally, and those over a long distance. Okay? And each of these steps share kind of similar attributes, but they each share some different attributes as well. So, for example, in cells that are communicating cell to cell, like touching each other, we have what are called gap junctions. So, direct connections between the cytoplasms of two different cells, either through protein channels that are connected to both cell membranes, or in plant cells, they have virtual, like literal holes in their cell walls that connect the cytoplasm of two adjacent cells, and they're called plasmodesmata. So in this case, it's simple diffusing signaling molecules between the two, either through the plasma membrane or the plasmodesmata, to communicate. Or you could have surface receptors like this. Think of like the lock and key type of thing that can send information like in the immune response for like a pathogen in a white blood cell that is based on contact. One receptor and one receiving the signal from another surface protein from the other cell. Okay? We'll talk more about in next semester about the immune system, about how this works more specifically. So kind of like we talked about, plants use plasmodesmata to send signals, and they travel through those little holes between the cytoplasm of the two adjoining cells. Local signaling, on the other hand takes place over a short distance. So, for example, in cell-to-cell recognition, recognizing, hey, you're another cell like me, so we should do this specific thing. Like we talked about over here with this example, there are surface molecules that can aid in that, like glycoproteins and glycolipids. If you remember us talking about in the cells unit, those different parts of the cell membrane and how those carbohydrate chains, we weren't going to bring up at the point, but they were going to be important. They serve as those surface molecules that can be recognized between different cells. And then there are other molecules or signaling molecules that can be sent over short distance, like growth factors, that are only stable and will deteriorate if they have to go farther than a short distance. And then maybe most famously, there's long distance signals. So they travel a large distance over the organism. So think like the nervous system, how the electrical impulses travel through neurons all the way from your brain to your feet, or how the endocrine system in animals uses like hormones to transmit messages from like your brain to your kidneys, or more properly like your adrenal glands, or from different places in your body. And plants also have hormones that are transported, some through their vascular system or through the air, that can signal between different plants or between the same plant to do maybe the same things like our endocrine hormones allow us to do. The the idea in local and long distance signaling, to talk more specifically, is that there is a similar process that occurs. A messenger molecule can be secreted or sent as from the signaling cell, the one that wants to stimulate another cell to do something. So, as an example, like here, it's called paracrine signaling. We're sending actual signaling molecules out. One cell secretes or releases a molecule that acts on a nearby target cell. So, for example, growth factor, like from the brain, is sent to different target cells that then respond by enlarging and growing more because of it. Or there's synaptic signaling, which we'll talk more about in second semester when we talk about the nervous system, where cells release chemical messengers called neurotransmitters that stimulate the target cell specifically. We're kind of going to lump both of these processes together and talk about a general pattern for local and long distance signaling in the signal transduction pathway. Here's another example like of long distance signaling. So in this example, you might have an endocrine cell. Let's say it's a pancreas cell, maybe a beta cell that's going to release insulin. And when the blood sugar rises to a certain point, it will release this insulin that then will travel to target cells to stimulate them to present GLUT4 receptor, which is glucose receptors on their cells to take in glucose. So that's an example of the signaling molecule being insulin. In general, though, if we want to generalize this to a three-step process, there are three stages for a signal to be received and something to happen because of it. And they're called the reception phase, the transduction phase, and the response phase. And it's summarized by this picture, which is also in your book. 1, 2, 3. So in the reception phase, the signaling molecule fits into part of the receptor, like a key fits into a lock. The signaling molecule, we have a fancy scientific name for it called ligand or ligand. Heard it both ways here. And it fits into the protein receptor. It binds with it, we say. That makes up the reception phase. So the signaling molecule is floating through the blood or through the air in plants or something, and will bind to the reception site in the receptor protein. So our target cell detects, quote unquote, that there's a signaling molecule coming from outside by the receptor binding with the signaling molecule, the ligand, the ligand. These receptors are specific to only specific types of signaling molecules, so only specific signals can bind to a certain receptor. You might have many different receptors for many different signaling molecules, but each one has high specificity. The next step is transduction. This is when the signal is being passed on and amplified to the place in the cell where something can happen to respond to that signal. So in this stage, we convert the signal into a form that can bring about a certain change at the cellular level. So one signal activated receptor can activate another protein, which activates another protein, which activates another protein. So like the game of telephone, all the way down to where we get a response. These proteins that become sequentially activated, like dominoes, are called relay molecules or relay proteins. And often this message is kind of transferred from one relay protein to the next through what's called protein kinases. And we'll get into them more in the next video when we go into the transduction phase more deeply. But just know that there are things that are being passed from one to the other to activate each. So here's an exact example of a signal transduction pathway where once the signal molecule is received, the ligand binds to the receptor here. That activated receptor ends up activating this molecule, all right, which then is able to activate this next molecule, which then is able to activate this next molecule, all the way down a chain of being activated by the last one, all the way till the last thing in the chain is able to do what this signal said to do. So why do we have all of these little domino links? The main reason is to amplify this signal so that one signaling molecule here can turn into exponentially more active proteins here to make a large cellular response. So one signaling molecule can create a large cellular response because each of these active proteins along the way can activate a large number of the one next in the chain before they deactivate. So our signal is growing exponentially on down the line. The last step that we'll talk about is the response. And this is what the cell actually does with the information that was being received and transduced. So after the signal that was passed through the transduction pathway, it triggers a specific cellular response, like an enzyme to do something or the activation of a gene. For example, here we have the reception, the transduction, and the response is that the last molecule in this relay pathway ends up activating transcription of a specific gene to turn it into mRNA and then a protein. So a lot of the responses are to start or to stop transcription of DNA into proteins. All right. Yeah. So lastly, there's a high specificity of the signaling systems within different cell types. So for example, liver cells and heart cells do not respond in the same way to the same messenger, like epinephrine, because they have different collections of proteins. So the same signaling molecule, when it attaches to a receptor on a liver cell, will cause a different response than perhaps that same signaling molecule when it activates a receptor on a heart cell, because they need to do two different things because of that signal. So even though the signaling molecule is the same, everything downstream, the transduction, the response might be different between those cells, so they can respond differently to that same message. That's all I wanted to talk about as far as this first video, just an overview of the signal transduction pathway. Next time we'll go into more detail with the transduction step. All right, we'll go into more detail with this and talk a little bit more about the transduction step.